Well, good morning everybody, and I certainly hope that you're ready for a big day, because I know that I am, and I know that we've got some really good things on the program today. So I'm not going to take any time. We have had one change in the program, that we have a new speaker this morning. Unfortunately, Scott Bell wasn't able to be here, but we have an incredible replacement. We have Mark O'Carroll. Mark is a respiratory physician at the Auckland City Hospital. He's the Clinical Director of Respiratory Services at the Auckland District Health Board, and he's the Director of the Auckland Adult Cystic Fibrosis Service. And his subspecialty roles include interventional bronchoscopy and lung transplantation. And we're lucky because he has a particular interest in medical education and is the clinical lead of the Northern Region Training Hub, and President of the New Zealand branch of the Thoracic Society of Australia and New Zealand. So we've certainly found an incredible replacement for one of our speakers. So I'm not really going to take any time apart from welcoming Mark, and Mark's talking today about Pseudomonas, where do you get it and how can you avoid it. So thank you, Mark.
Right, well, thank you. So Scott, unfortunately, couldn't be here, but so he's given me his slides, and I'm going to endeavour to walk you through his slides and interpret them. Some of them are pretty complicated, and I struggled with them, but I've spent quite a lot of time talking to them. It's worth just acknowledging some of the work that Scott and his group's done, particularly in this area. I had the good fortune to work as his fellow actually 10 years ago, and did some work in Pseudomonas then, and that really was just the start of a whole series of publications that Scott and his group have subsequently published. And what I think you'll see from these slides is that they're basically gradually unravelling the story and gradually working out a lot of really interesting things about Pseudomonas. I've put one slide in at the end whereby I've tried to summarise it all, and I think the key thing that you should take away from this is it's very, very complicated, and there are very many different factors that contribute to Pseudomonas in terms of potential sources of it. Try that one. So the talk's split up into 7 different parts there, and what I'll talk a little bit about initially is the epidemiology, talk a bit about Pseudomonas and the impact it has in cystic fibrosis, and this audience will be very familiar with that. But talk a little bit about the literature which highlights the impact of Pseudomonas in CF. Then talk a little bit about the origins of cystic fibrosis in relationship to, the origins of Pseudomonas in terms of the environment. Touch on some of the work around shared strains that have been increasingly recognised, whereabouts they've been recognised and the significance of them. Talk a little bit about Pseudomonas in terms of the biology, how it actually has an impact in the person with cystic fibrosis. Then walk through some of the work that Scott and collaborators have done, which has sampled various different environments throughout Australia, and some very interesting results looking at the origins of Pseudomonas there. And then touch a little bit at the end on how transmission might be occurring, where we're at currently in terms of our understanding and knowledge, and the future direction of research, and then the implications for all of this for us collectively in terms of how we basically coordinate our care, how we look after people with CF, and what you need to do. So Pseudomonas, we know that Pseudomonas is ubiquitous, it's everywhere. We know that there are many different sources in all sorts of different environments. We know that it's a pathogen not just in humans and not just in cystic fibrosis, but also in other animals. and a broader range of different animals. And we certainly know that it's a big problem for the person with cystic fibrosis, and it's the most common cause of lung infection in CF. This data is from the US and taken from the CF Foundation Patient Registry. The point really is just to follow the red line, which just shows the prevalence of Pseudomonas aeruginosa infection in relationship to age across the x-axis. So you can see that that from a young age, Pseudomonas rates are relatively low, but as you move through into adulthood, the percentage of people with Pseudomonas increases progressively. So we know that from the US, that it's the dominant pathogen. We know similarly from data from the Australian CF data registry, this is from the 2009 data registry published in the Medical Journal of Australia in 2011. The red circle is not quite in the right place. It should be down a little further. And if you just look at the line there of Pseudomonas species and Pseudomonas aeruginosa, you can see the rates of Pseudomonas infection in children, adolescents, and then adults. And you can see that progressively gets greater. So that's well known and well documented. We can see it in the US population, and we can see it in the Australian population. This is a bit of data from the Prince Charles Hospital, which is one of the biggest CF centres in Australia, from Scott Bell's centre. And the three different plots there show the rates of Pseudomonas infection over time, over a period of the past decade. So at the top you've got your mucoid and non-mucoid Pseudomonas, so your two different types in terms of how they look on a plate, and at the bottom you've got patients who don't have Pseudomonas infection. And what you can see, if you look at between 2008 and 2010, there seemed to be an increase in the number of patients in the clinic who didn't have Pseudomonas infection, and a decrease in patients who do have Pseudomonas infection. So it's trended down and then remained stable over the last 3 years. And that's probably related to a number of different things. Quite a big part of it relates to the fact that we're starting to see, particularly in the Queensland group, more patients transitioning without ever having had Pseudomonas infection. So quite a lot of it is the transition of a cohort of patients who actually have a lower rate of chronic Pseudomonas infection. Part of it also probably relates to infection control practice and changes that have taken place in infection control. So there's quite a significant difference there, and you've gone from around 70% in 2008 to around 60%, and that's just one centre. So can't read too much into that, but it's just a bit of local context. So we know it's the dominant pathogen. What about the significance? It's well documented the impact of Pseudomonas on CF disease progression. So listed there on the left a list of publications which have effectively shown this and demonstrated the association between chronic Pseudomonas infection and disease progression, and you can see there are lots of them. And we get similar messages from the data registries which show that Pseudomonas infection is associated with more advanced disease, with more admissions, and with deterioration in lung function. And the reasons for that seem to be that Pseudomonas is associated with a greater host inflammatory response, and it's thought that that host inflammatory response is damaging to the CF lung, resulting in an accelerated decline in lung function. We can clearly see that those people who have Pseudomonas infection, when you compare it with those who don't, have a greater frequency of exacerbations and increased treatment requirements. It's associated with a reduction in quality of life, and it's clearly associated with a reduction in survival. So where does it come from? Well, historically the paradigm's always been that we have thought that Pseudomonas is acquired from the environment. And so the historical paradigm that we've always operated on has been that children acquire Pseudomonas from the environment at some point, and there is certainly evidence historically that supported that showed that there was a diversity of strains, of unique strains of Pseudomonas in young patients, consistent with the supposition that Pseudomonas is environmentally acquired. And there were early reports which showed that the prevalence of shared strains, so two people with CF having the same Pseudomonas, seemed to be confined to those people who had very close intimate contact, so classically siblings or others who have close intimate contact. And that was sort of the starting point of what we thought historically with chronic Pseudomonas in terms of the acquisition of it. And the thought basically being that at least in siblings, sorry, jump one forward, at least in siblings, Can I just go back a slide, please? And one more. Yep, thanks. In siblings, it was thought there were two main possibilities. So it was either thought they were sharing it, so basically with close contact transmitting from one sibling to another, but equally those siblings living in the same environment may well have acquired it from exactly the same environment, but living in the same place, exposed to the same environmental Pseudomonas, and therefore acquire the same Pseudomonas strain. What about shared strains? So this has really been something that's unfolded in the past decade. We now recognise the fact that there are shared Pseudomonas strains really in CF patients throughout the world, and there have been a lot of publications on this. So if we look in the Northern Hemisphere, there are well-described strains. The earliest ones were the Liverpool epidemic strain, so the LES is a Liverpool epidemic strain, and closely following that was the Manchester epidemic strain. And subsequently there's an array of others both in Europe and in Canada, where they've described the Liverpool epidemic strain present in Canada. And then in Australia there have been a series of publications, some of those listed there on the right, which again describe shared strains. First of those in Australia was at the Royal Children's Hospital in Melbourne, with reports in the early 2000s, and that strain has now been labelled as the OST1 strain. And there are a series of other major strains, OST2 being the major Brisbane strain, and OST3 being the major shared strain seen in Tasmania. What I mean by shared strain is I mean that it basically is exactly the same strain of Pseudomonas harboured by more than one patient in a given CF clinic. It's exactly the same based upon genetic fingerprinting. So you type them using a variety of different methods, but when you look at the genetic fingerprinting, they are exactly the same. What comes out of that work is we can see major shared strains whereby they're shared by quite a large number of people, but we also can see minor shared strains, so where they're harboured by a much smaller number of people. The evidence points to the fact that these shared strains have a detrimental effect. And the first and strongest pointer to that was from the Melbourne group and the Royal Children's in Melbourne, whereby the description of a shared strain amongst shared strain amongst 5 children who went on to have premature death. They all had the same strain of Pseudomonas, and it was genetically identical. And clearly that was the start of a lot of this work, and which led on to much further investigation as to the significance of these. Beyond that, the other shared strains that have subsequently been reported and followed have not been associated with premature death. And there have been varying results in terms of the significance of them. So some have demonstrated that the presence or the presence of a shared strain of Pseudomonas is associated with increased treatment requirements, with greater decline in lung function and nutrition. And the Liverpool epidemic strain is is probably the most significant of those. The Manchester strain's different in that the group of people harbouring the Manchester epidemic strain had increased treatment requirements, but they didn't have accelerated decline in lung function, and they didn't have any difference in survival. So the signals from that are, clearly a real concern around that first description in the Melbourne group, but subsequently what we've seen is that the shared strains seem to be associated with greater treatment requirements, a bit of a signal that some of them are associated with detrimental effect on lung health, but other indicators that they don't necessarily have a significantly adverse impact on survival. Coming out of that early work really led Scott and his group together with collaborators throughout Australia to develop this study. And this is a study that was designed to look at the true prevalence of all shared strains throughout Australia. So all of the studies prior to that had been isolated to sort of single centres, whereby each centre would look at their patients with CF and determine whether the shared strain was present within their own clinic. This looked to see whether, take a more global view of Australia as a whole, and determine the prevalence of shared strains throughout the whole of Australia. And so you can see there that 18 of the 21 centres in Australia enrolled patients, 22 different labs throughout Australia. There were about 900 patients that ended up submitting sputum samples for this study, and in total they sampled just under 3,000 Pseudomonas samples. So they took each of those Pseudomonas bugs and they genetically fingerprinted them. And basically just took standard sputum samples, grew them in the lab, picked the Pseudomonas ones off the plate, and then they genetically fingerprinted them. So the bits at the bottom here are the scientific methods, and in essence this scientific method here, the PCR-based thing, was a test that's been developed in Queensland. It's a quick test to be able to identify related or identical strains, and then this here, the MLST test is the gold standard test, so it's recognised as being the best test that we've got to fingerprint things. So they screened with this and then they did the gold standard test. So they used techniques that basically are indisputable in terms of the results. And then they looked at an analysis at the end in terms of the relatedness of these things, and this starts to go beyond my level of understanding of Scott's slides, but I'll show it to you. So these are the results, and the results are a bit sobering, to be honest, when you look at the significance of this problem. So you can see there that there are 983 patients, that 38%, so 373 out of the 983, simply had unrelated strains. So they had, when you type their Pseudomonas, they were unique to them and not shared with anyone else. 610, so just under 2/3 of patients, shared a strain with one other person. When you look at most of those strains, a lot of those, so there were 38 shared strains. I'm talking across the entire of Australia. There were 38 shared strains. Now, half of those were strains shared between two people. So again, comes back a bit to what I said at the beginning, the sibling type thing, or people with close contact, and you can explain it from that perspective. Can't explain it from that perspective for a lot of the rest of what we found, which is we saw 14 strains shared between 3 and 19 patients, and then we saw 5 strains in much bigger clusters. So the 5 other strains listed there, and they're labelled as the Australian 1 through 6 strains there, shared between 31 and 220 patients. And if you focus particularly on OST1 and OST2, you can see that of the 18 centres, CF centres that were involved in the study, the vast majority had patients in them who had either OST1 or OST2. So it shows that we've got shared strains of Pseudomonas that are ubiquitous throughout the population in Australia. This is just a different way of representing the same data, and the blue bars are the OST1, the red bars are the OST2. The percentages basically are the percentage of patients harbouring either OST1 or OST2. So 22% of your total patients, of your 983 that are involved in this study that basically ran from 2007 through 2009, a fifth or just over a fifth of them had the OST1 strain, and just under a fifth had the OST2 strain. And it was different in different clinics. And so each of these across the x-axis here are different CF centres in Australia. And you can see some here predominantly had the OST2 strain, particularly the Queensland centres, whereas others over here, Victoria particularly, predominantly had the OST1 strain. And there were some whereby you could clearly see a significant difference in the presence of one versus the other. And there are two different Queensland centres. So it's different for different centres, which makes it slightly more complicated, and then trying to think about why that is. When you then plot out those major shared strains and you look at where people live, what we could see was that the majority of patients with the OST1 strain lived in southeast Queensland. So there were 220 of the patients, 220 of the 983 harboured the OST1 strain, and 3/4 of those live in southeast Queensland, southeast Australia. So each of these different things are your CF centres. So your Brisbane group, your Sydney and your Melbourne groups here, and the height of those different bars represents just simply the total number of patients who are found to harbour the clonal strain. So the blue bars being the OST1. So we see OST1 particularly over here in Australia. When you look at OST2, it seems that it was predominantly in Queensland and Western Australia. Now recognise that we didn't get every single centre was not sampled here, but the vast majority were, so 18 out of the 21 centres in Australia. There were some other observations made when you look at the shared strains and, in terms of factors that were related to it, and probably the most significant one I'll draw out is the impact of age. So if you look at the pie graphs in the bottom right here, and we take children under the age of 18, the green part of the pie graph are those harbouring a unique strain. So their strain's their own one, it's not shared with anyone else. So half of the children had a unique strain and didn't have shared strains. If you compare the size of the green in that pie graph with those over the age of 18, you can see that about a third of adults, compared with a half of children, have unique strains, and many more adults shared strains. And you can see that those shared strains, the red is OST2, the blue OST1, and then the grey bar is other shared strains. So 2/3 of adults shared a strain. So age is important. We see that as people get older, they're more likely to harbour a shared strain, and the rest of what's going to follow will explain some of the reasons potentially why that is. We basically see that stringent infection control probably has an impact, and that slide I showed you right at the beginning of the Prince Charles rates of Pseudomonas infection dropping probably has something to do with that. And then I've touched on the difference between adults and paediatrics. This just basically, these plots look at some of the other strains in terms of where they come from, where they're prevalent within Australia, and the distribution. So the plots here really are of each of these dots being people with shared strains, the colour obviously representing the nature of the shared strain, and then where they live throughout Queensland. So you move on to saying, where does it all come from? So is the reason why we're seeing a lot of shared strains in people with cystic fibrosis because they're acquiring it from the same environmental source. And so it led into the next bit of work that, that Scott and his group did, which really sought to look at the biodiversity of Pseudomonas in the environment. And the hypothesis was that we're seeing a high prevalence of the OST1 and the OST2 strains, and another one, the OST6, in the Queensland CF centres because they're there in the environment. And so they basically designed a study, designed a study to answer that question. So is it basically that all of our patients in Queensland particularly were acquiring it from the environment and it's shared just because it's everywhere in the environment? And so they went about sampling a wide array of different environments. Okay, so sampled patients with cystic fibrosis, took patients without cystic fibrosis but who had other lung disease, so bronchiectasis, and other sorts of infections, sampled animals, and then the environment. And in this work, they went about and sampled all sorts of different places, went out and sampled various parts of the Queensland River, and then they typed them again using that gold standard test, so the best test that we've got in terms of genetic fingerprinting. And this is what they collected. And so the niche basically being where does it live, and then your different sources down the left-hand side here. So those three different ones, and then these are what they got out of their isolates. So in total, in sampling all those different sites, got 501 different isolates of Pseudomonas, and they typed them. And so we work our way down the slide and start at the top. Here you have CF patients, sampled 160 specific isolates, found that 60 of them were unique, and found that 98 of them were either major or minor shared strains. Then took non-CF patients, sampled 129, all sorts of different sources, and then sources in animals, all sorts of different animals. Well recognised that horses particularly are prone or an ideal vector for Pseudomonas infection. And then the environment, so all sorts of different places, looked in homes, looked in water sources, looked in pools, rivers, Brisbane River, Logan River, so on and so forth. So what they find, when you look at the Brisbane River, sampled 46 sites and 2/3 of them, basically they grew Pseudomonas, so isolated Pseudomonas from 2/3 of the sites they sampled, and in basically each of those they found a large number of different colonies, ranging from one particular site from just having a single type of Pseudomonas to some sites having up to 25 different types of Pseudomonas in a single sampling site. And they found that when they typed those, and that's the screening test there in terms of doing the genetic fingerprinting, it ranged from one unique type through to 6 different types of Pseudomonas. And they then took those unique types detected by the screening thing and went on and did the gold standard fingerprinting test. This slide tries to summarise those results, and it's complicated, but it illustrates the point very nicely. So what we saw is 274, and ST is a strain type, so they're unique types of Pseudomonas. And the plot here basically represents the different niches. So this is a veterinary source. Okay, so from animals is the yellow oval there. The green one are patients without cystic fibrosis. The red one there is patients with cystic fibrosis, and then the purple one is from the environment. The overlapping and the intercepting part basically represents strain types of Pseudomonas found in more than one niche. So if we just focus on the veterinary one here, for instance, you can see there were 79 unique strain types of Pseudomonas found in animals. 53 of those were unique, so they were unique to animals. They were found just in the animal vector, not found anywhere else. However, when you look at the overlapping, you could see 8 of these that were found in animals were also found in people without cystic fibrosis. So they were present in animals, but they were also capable of causing infection or disease in people without cystic fibrosis. These 5 here were found in animals, but they were also found in people without cystic fibrosis and found in people with cystic fibrosis, right? So they're found in 3 different niches, 3 different parts of the environment: animals, people without CF, people with CF, and so on and so forth. So these 5 here basically found within animals, but also found in the environment when we sample various water sources throughout the environment. If we go to the bit we're interested in particularly in terms of the CF population, we can see that we had 90 strain types found in our 160 patients that we sampled with CF. 60 of those were unique to the person with CF. 7 of those were found in people with CF and in people without CF. 3 of them were found in people with CF and from the environment, and from people without CF. So you've got there all those intersecting lines there, so found in 3 different niches. And you can go through and analyse that and see that what you've got is you've got quite a lot of Pseudomonas ubiquitous throughout the environment, and quite a lot of it's present in more than one place. What does that tell you? It tells you that Pseudomonas is capable of adapting to different environments, and the degree of adaptability is variable. So some Pseudomonas basically will adapt to a given environment that doesn't translate to others. Other Pseudomonas basically has a much greater adaptability and can live in more than one niche. You can see there that summarises that 28 of the CF strain types, so 31%, almost a third of the strain types found in people with CF were found in another place. So it supports the initial contention that at least some of the Pseudomonas we're seeing in people with CF is environmentally acquired, and by environment I mean the water in the environment, but I also mean animals, and some of it also comes from people without CF. This slide looks at the shared strains of Pseudomonas, and the key point to take out of it is that the major shared strains, when you look at OST1, OST2 and OST3, so the main ones that we found in the original Australian CF study where they sampled the 18 centres, and it says those major shared strains, do we find them anywhere else in the environment? So are they found in another niche? The answer to that's no. So OST1, 2 and 3 were found in CF patients only. So if we go back to the beginning of the study, the hypothesis was that we're seeing a high rate of OST1 and OST2 infection in patients in Queensland because they're all getting it from a common environmental strain. The answer to that is there is no evidence that that's the case. So the OST1 and the OST2 and OST3 for that matter, these major shared strains were not found in the environment. So the only niche where you found those are in CF patients. So the way to interpret that is to basically interpret that as that particular type of Pseudomonas has adapted in a unique way to then only live in the niche of the CF host and not live anywhere else. Also means it's also got implications for then where do people get these major shared strains from, and they get these major shared strains from other patients with CF. And that's the key takeaway message with that. So the hypothesis that was put forward in this study was clearly disproven and shown that basically that they're not getting these major shared strains from the environment. There are a whole lot of other minor shared strains, and there's a lot of work and a lot of publications which have looked at these minor shared strains and that have been found in more than one niche. And if you look at publications about these, they've been described in all sorts of different places. So the same types, often with these minor shared strains, these same Pseudomonas isolates and these unique strain types have been described in Australia, but equally they've been described in Europe and North America. in CF patients, in the environment in non-CF patients. So many of these more minor shared strains are in fact ubiquitous throughout the world. And so you're not just talking about the ones you get in Australia being unique to Australia, you're talking about them being common environmental sources. And there's a lot published about many of these minor shared strains, and I won't go through all of that. Some of them, are particularly, well described in veterinary medicine. So ST883 is something that's only detected in horses and thoroughbred horses. So again, it comes back to just the way you think about that is that particular strain type of Pseudomonas is adapted so that it basically only lives in the niche of the thoroughbred horse, and it's not found anywhere else. So what that tells us about the ecology of Pseudomonas that we're seeing in CF is that we see the same ecology of Pseudomonas in veterinary medicine and other animal sources. So it tells us about the nature of the bug, that it basically adapts and then will only be present in one given environment. So we're seeing that, and that's a good example of it. And you could say that what we've there seen in horses, potentially we've seen the same thing with our OST1 and OST2 in the setting of CF. And these are other strain types, and you can see that they've all got different numbers, different names. The significance of them is that they're unique, but they're described in all sorts of different niches throughout different parts of the world. So here, ST274 is a unique strain described in Europe, also described in non-CF patients. ST155 you can see described in the setting of CF, non-CF. The environment, so seawater samples from Japan, and animals causing bronchopneumonia in dolphins. And so that particular type of Pseudomonas still retains the ability to grow and cause infection in a variety of different niches. The Australian CF-BAL study gives us further insight into the nature of Pseudomonas and where it's coming from. So you'll be familiar with this study because it's been going for quite some time. And the idea of the Australian CF-BAL study was that it was a randomised controlled trial designed to determine whether doing BALs, so bronchoscopies and BAL samples on a regular basis, and using those results to direct therapy would lead to an improvement in outcomes. So, and basically the control group had standard oropharyngeal or cough swabs and therapy directed based upon those culture results. So it basically says, if we're more aggressive and sample more directly, does it result in an improvement in outcomes? I'm not going to talk about the results of that study, but what I'm going to talk about is what we've got out of it in terms of the Pseudomonas samples that have then been isolated either from the BAL samples or the oropharyngeal samples, the cough swab samples, because we're then able to take those and type them. So if you take those samples, Pseudomonas aeruginosa samples, 81 children in the Australian CF-BAL study isolated Pseudomonas either from the BAL or these are oropharyngeal samples, cough swabs, and took all of those and then were able to get 70% of them and genotype them, do the fingerprinting test. And Tim Kidd did this work with Scott and Claire Wainwright, who's been the lead author with the Australian CF-BAL study. What they saw in this paediatric group of patients is that they only had one patient who had either OST1 or OST2. 2/3 of patients had strains that were indistinguishable from strains detected in other niches, environment, non-CF patients, animals. And some of those were the more minor shared strains I've talked about, so the ones that I showed you before that have been described in a variety of different environments in all sorts of different places throughout the world. The ARREST CF study is something that was initiated in Western Australia, and there's a Melbourne site. We've been able to basically get some more insight about Pseudomonas from the Melbourne ARREST CF study. The aim of that was to determine whether there were geographical differences in the acquisition of Pseudomonas in young people with CF. So what I've shown you is that you can get CF from the environment, so you get Pseudomonas from the environment, you can get Pseudomonas from other people with CF. I've shown you that in young children they're more likely to have environmental strains or strains that are detected in other niches, whereas as you get older and adults you're more likely to have shared strains. This analysis was designed to then look at the Pseudomonas isolates in young children to look at where they lived in relationship to the likelihood of acquiring Pseudomonas, and investigate the relationship of their isolates to other isolates from other niches. The bit that we're interested in this slide is the group of people with Pseudomonas. So we had 25 patients in the Melbourne ARREST CF study that grew Pseudomonas on BAL. And interestingly, 19 of those 25 lived in came from a regional centre, only 6 from a metro centre. So if you lived in a rural place, you seem to be more likely to get Pseudomonas in children. These 9 were Pseudomonas on sputum, so or cough swabs, and half were from regional and metro centres. That plot there basically shows the geographic distribution in terms of where people live when you type the first Pseudomonas that they acquire using the BAL samples. So again, taking those 25, and we saw that they were much more likely to come from a regional centre, suggesting to us that the first acquisition of Pseudomonas in children is more likely to be from the environment and more likely related to greater exposure to the environment. When we then type those, so again the MLST is the genetic fingerprinting, and we type those BAL samples, we saw that none of them were OST1 or OST2, and that 10 of the 15 were seen in those other niches we've talked about. So again, supporting the fact that they're acquiring it from the environment. And the strain types detected again were some of those minor shared strains that I've shown before, again from different niches. This is an awful slide, but because it's very hard to explain. What it's meant to demonstrate is the relatedness of strain types of Pseudomonas. So the way in which you do something like this is you need a scientist first. But the scientists, what they do is they take the Pseudomonas and they basically pick 7 different what we call housekeeping genes within the Pseudomonas. So take all the genetic material. There are certain parts of the genetic material that we all have that are conserved over time, and they take 7 different genes and they type them. And basically, if all 7 of those are exactly the same, then that ends up being 1 dot. If basically one of them is different, then there's a line, so they're related. So you've got 6 which are exactly the same, 1's different, then they're closely related. So it's kind of a way of saying, let's look at all the Pseudomonas that's in the environment and try and plot it in a way which shows how closely related they are. And the message you get out of this is that when we look at OST2 and its relationship to other common environmental strains, we can see that here's your ST883, so the strain type of Pseudomonas only found in thoroughbred horses. And so you can see what I've shown you is that OST2 is found only in people with CF. I've shown you that ST883 is found only in horses. And whilst they're not directly related, they're closely related. So it suggests that they're related in a way whereby they have a characteristic which allows them to adapt to one unique environment and then only remain within that environment. So it's a way of trying to demonstrate relatedness. So what else? What else can we look at here, or what other factors might be important in terms of the acquisition of Pseudomonas? And there's emerging evidence that the environment matters. And by that I mean there's emerging evidence that climate determines the bugs that you potentially get exposed to and the bugs that you potentially get infected with. Now in CF, the best example of that is Burkholderia pseudomallii. So Burkholderia pseudomallii is a bug that's found particularly in tropical climates, and it's a big problem in Northern Territory. So if you basically go up to Darwin, you'll find that malleoidosis is a big problem, and it's a very common cause of pneumonia in people without CF. But we tend to see that Burkholderia pseudomallii doesn't cause infection in areas elsewhere in Australia, so you don't tend to get malleoid infection anywhere else. And that relates to the fact that it's in the environment, and it lives in the environment in the tropics. And there are well-described cases of exposure where people with CF will go to tropical regions, so the Northern Territory in Australia, and then Thailand, tropical regions there, and potentially that's a source of exposure. And there's a bit of evidence that similar things exist with other bugs in CF, Cepacia, and there's a bit of evidence now emerging that suggests the same thing may exist to some extent for Pseudomonas. This really just demonstrates that in terms of the difference in the prevalence of particular bugs in tropical Australia versus subtropical Australia. And just draw your attention to two things here. So this is Burkholderia pseudomallii, 7.2% versus 0.6% in subtropical. And in non-tuberculous mycobacteria, so a different sort of bug that causes problems in CF, again, much more prevalent in tropical Australia than in subtropical. So climate seems to matter in terms of exposure to bugs. There are other factors about the climate that are probably also important when we look at other bugs as well. And this plot here looks at the incidence of Burkholderia cepacia infection over time, and you can see dating back over 10 years. And from 2005 onwards, there seemed to be a significant difference in that the rates of Burkholderia cepacia infection went up significantly. So going from about 0.5% to around 2%. And when you look at that and say, well, why might that be? There seems to be a correlation between the rates of Burkholderia cepacia infection and total rainfall and something called the Southern Oscillation Index, which really is just a measure of humidity. So environments whereby you have more total rainfall and more humidity seem to be associated with greater rates of Burkholderia cepacia infection. And the same thing is now starting to be suggested with Pseudomonas. And the evidence would suggest that the closer you live to the equator, the more likely you are to acquire Pseudomonas. And that was borne out in the Australian CF-BAL study, which shows that if you live further away from the equator, you have lower rates of Pseudomonas infection. So, for instance, the rate of Pseudomonas infection in the children who live in Auckland in the BAL study was significantly lower than what you saw in Queensland. So distance from the equator or climate seems to basically determine or perhaps be a determinant of how much environmental exposure you get to Pseudomonas from those other niches. These are complicated slides, and the key point I think to take out of this is these are quartiles, and the different quartiles basically show the temperature of the environment, generally speaking, and these rates show freedom from infection of Pseudomonas. So you can see that in the coolest climate, those people had the lowest rate of acquiring Pseudomonas, compared with the hottest climate, had the highest rate of acquiring Pseudomonas. Still hard to rationalise the whole story though, and this has helped us a lot in terms of understanding some parts of it, but it hasn't helped us understand all of it. So we know that children do harbour environmental strains, yet some of them also have shared strains. So what else might influence that? Well, there's no doubt at all that the social networks, in terms of the complexity of social networks, have a part to play. This work was done with scientists who do work particularly around the likes of avian flu, and they look at the migration patterns and where people move and their relatedness in relationship to how bugs might spread. And the graphic there is sort of meant to simply demonstrate the concept of a relatively simple network whereby each of these dots are individuals, and you've got a simple network whereby most of them don't have anything to do with one another, and there's not a lot of overlap, and so the connectivity is low. Whereas this here is an example of a much more complex network, whereby there's a lot more overlap and therefore a lot more contact and a lot more potential for cross-infection. When you look at the migration patterns and the movement of people with CF, in many respects it provides the recipe for a perfect storm in that it's a complex network. So this was part of the Pseudomonas prevalence study in Australia. And what they did when they did that study, so basically everyone that was recruited in, so 983 patients, they took a history from those patients, asking them where they'd ever been cared for. So any point of care previously. So obviously their primary CF centre was one node, but then they asked them where they'd been previously. So had they travelled, had they been at any other centre in the world, had they been in another CF centre, but also had they been in any other hospital. So regional centres, and then tried to look at the relatedness or the complexity of that, really to basically explore the fact that this may be a determinant or a way in which people are sharing strains of Pseudomonas. And so the nodes that you've got here are your CF centres, so you've got your 18 different CF centres, and then you have 67 other treatment centres, and this is a plot. And effectively the lines join the dots of where people have been. Basically each of those lines really describes patient movement between nodes. 515, so remember 983 in the study, 515, half of them had contact with another node. 126 had been in 2 other places. 20 had been in more than 3 places in terms of healthcare settings. 861 total ties. And so you can see there that it's a complex network and there's a high degree of connectivity. And when you plot it out like this graphic demonstrates it. So in the children, there was much less movement, whereas in adults, naturally there was a lot more. Of course, any adult would have transitioned from a paediatric to an adult clinic, and by nature of that, it's always going to be much more complicated because they've been in 2 nodes, but they also were much more likely to have been in some other healthcare setting. And that might be as simple as going to the local regional hospital to have IV antibiotics for a period of time closer to home. So we saw much greater mobility amongst adult patients compared with children, which should come as no surprise. It's common sense, but it relates to the fact that there are lifestyle changes, that people move, that children are much more likely to stay with the nuclear family, at least initially, but then as they grow up, go to university and work relationships and travel, then they're going to basically have much more exposure to those other healthcare settings. And what we saw is that's the relationship there between seeing a lot more shared strains in adults. How does it happen? So if I'm saying to you that one of the mechanisms is transmission of these major shared strains from one person with CF to another, then how does it happen? What is the mechanism of cross-infection? And this is an area of active research. So historically, the 2 metre rule was based upon the premise that when you cough, you aerosolise droplets, but those droplets are big, and the big droplets will drop to the ground within a 2 metre radius. And that's basically been the historical principle of what we've operated on. The evidence regarding that is not particularly good, and there are 2 studies which have been reported which have shown short-term contamination of air in rooms for patients with clonal Pseudomonas infection, and that's the concern, and that's clearly what we're concerned about. This is a way in which you try and demonstrate that or try and scientifically evaluate it. So these are historical experiments, and what you've got in here is you've got a series of plates, these Anderson plates, Anderson samplers, and you've got basically the person coughing into it, and the plate that it settles on, it gets filtered out. So you filter out your big particles first, and then the smaller ones will keep travel through, and so each of the plates basically will detect a different particle size, and this is what these different stages are. So these are very, very tiny particles, and these are the much bigger particles. When you look at the plates basically after they go through the filters, you can see that they're all growing Pseudomonas, and so it argues really against the hypothesis that they're all big droplet particles and they're all going to drop and not go any further, because if that was the case, then they'd be on one slide and you wouldn't see them on the rest. And that really just demonstrates it, that these are colony forming units, so basically the density of Pseudomonas on those plates, and you can see with each of those stages on your Anderson plates, yeah, there are different numbers, but they're there. And so they're not just all stage 1 big droplet particles. Clearly the key question is how far do they go, and how long do they stay airborne, and do they remain viable? And that's the subject of ongoing research that's currently been done in Queensland by Scott's group, and also in the US. And so there are 2 studies, experiments, basically trying to address the questions of distance. How far is it? How far, should we consider it to be safe? And how long potentially do those particles remain in the environment, basically both on the surface but also in the actual air. Can't give a talk about the impact of shared strains of Pseudomonas and talk about the infection control issues without thinking about the impact that has had on patients with CF. We've basically gone from environments which were friendly and a point of social contact for many people with CF to now being a much more isolated environment. It's not impossible, and some of the environments that I've been in, the Manchester clinic for instance, with social networking can still allow contact, but it has had a big impact. These are a series of quotes that came from Andy Jones. This is Andy Jones basically, who now runs the Manchester Adult CF unit, and basically these came after they moved to change their infection control practice so that there was no intermingling in their clinic spaces or in their hospital environment at all. And they're the sorts of emotions that you'd expect, and it does have significant implications. Also has significant implications for how you then run your clinic. So if we're then going to say that we want to basically isolate or conduct infection control practices whereby we try and maintain each type of bug away, isolated or removed from other people harbouring different types of bugs, becomes logistically very, very difficult. So that there is the schedule of Scott's clinics in Queensland. And that you can see there that they're busy and that they basically have a very complex schedule of when you see patients on what days. Of course, you then need to build into that, well, where do you evaluate and when do you evaluate someone who's acutely sick and needs to be seen, and how you then go about organising our CF services. So there are real challenges for us here, and we've made some inroads, but we've got a lot of work to do. This is an important slide, and the significance of this slide, firstly, is that it's old. So it's not based upon recent data, just about finished. And so you can see here 1998. And what it did is it looked at 3 different cohorts. This is a UK cohort, and it took basically patients who had all their CF care in a CF, so had all their care in a CF centre, compared those who had just had their care in an adult centre with those who came into new referrals into an adult centre who'd never been cared for in a CF centre. And there are some important messages here. Those who had care in a CF centre for their entire time had a significantly better FEV1. So they had significantly better lung function. This is the statistical significance, so it's significant. 61% versus those that had never been there. They had better nutrition, BMI of 21.6 versus 18.3. However, look at their Pseudomonas colonisation rates. 90% versus 72%. Again, statistically significant. I don't take out of that, don't go to a CF centre to have your care. But we need to look at that and see what it's telling us, and it does tell us that people are acquiring Pseudomonas and have historically acquired Pseudomonas from other people with CF. They're still basically doing better as a result of CF care, no doubt about that. There's no doubt about it. So the challenge for us is to continue to basically address this in terms of the potential to acquire Pseudomonas in CF clinics, so that we'll then see even greater improvements in these things that really matter, clinically meaningful outcomes. So this is the only slide I put in. Everything else I've shown you is what Scott did, okay? And all I've spent is trying to understand his slides. So if we summarise it, I think the bottom line is that Pseudomonas is everywhere. That the nature of the CF lung is that basically, it is the unique environment that Pseudomonas will grow in, and we know that certain strains of Pseudomonas will only grow in the CF lung. Acquisition in childhood is more commonly from the environment, no doubt about it. Where you live, the climatic conditions, they influence your acquisition. The complexity of social networks are important. They contribute, migration, movement, and a lot of that relates to exposure to other people with CF, but also relates to exposure to other environmental niches. Shared strains are almost universal in large clinics. There are some people who are still holding on to the belief that they don't have them. We don't get them in our clinic, and I don't think that we can believe that anymore. I think we have to basically recognise that they are universal in big clinics. The magnitude of the problem is going to be different in different clinics, and perhaps part of the approach to this is to start with where we think the big problems are. The work that I've shown you from Scott and his group's basically gone a long way towards helping us understand many of these things, and there's a lot more work to do, but I'd put it to you that we're a hell of a lot further ahead in terms of understanding it 10 years on than we were in the early 2000s. But there are challenges for all of us in terms of how we then organise and manage our CF centres so we can continue to basically deliver the improvements that we're seeing, continued improvements, but also limit the acquisition of Pseudomonas. Thanks very much.